Hi Nilesh, hi Parth. हाय अयान, गुड आफ्टरनून शुभम। गर्मी में बोइल्ड वाटर, यहाँ पर गर्मी नहीं है, इट्स 19 डिग्री सेल्सियस, 17 भी रहता है। इतना ब्लर क्यों है मेरा ये? बिकॉज बारिश हो रहा है। हाँ, बारिश भी हो रही है लेकिन अभी रुकी हुई है। वो टेरेस का पानी गिर रहा है ना, साउंड आ रहा है ना, वो वाला। मैं वाईफाई से जुड़ी हूँ ना, लेकिन फिर भी बहुत ही ज़्यादा ब्लर आ रहा है, पता नहीं क्यों। 60 मिनट का मैंने भी पिया गर्म पानी। मुझे तुम्हें देखने के बाद ही ऐसा लगता है कि तुमने मछली भात खाया। बात ये, तुम्हें कुछ हुआ था क्या? कल तुमने ऐसे करके पूछा ना सर्वजीता। मांस भात, हाँ वही। हाय निशांत। हाँ, कल मैं डिप्रेशन में थी। ओह, मैं भी होती रहती हूँ। हाँ, सारे एस्पिरेंट के रिश्तेदार डिप्रेशन आते रहते हैं। हाँ, तो स्टार्ट करें स्लॉट 1। मैंने लिखा तो है कि हम लोग 1 AM तक नहीं सोएंगे, लेकिन आई थिंक मैं सो जाऊंगी उससे पहले। स्लॉट 1 है, स्लॉट 1। ये लो, कर दिया है। शुभम। PCR, recombinant DNA technology, polymerase chain reaction, and enzyme linked immunosorbent assay ELISA. Enzyme linked immunosorbent assay ELISA are some of the technique that serves the purpose of early diagnosis. Presence of pathogen, bacteria, virus, etc. is normally suspected only when the pathogen has produced disease symptoms by the time the concentration of pathogen is already very high in the body. However, very low concentration of bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of the nucleic acid by amplification of their nucleic acid by PCR, amplification of their nucleic acid by PCR, amplification of nucleic acid by the PCR. Can you explain how PCR can detect a very low amount of DNA? PCR is now routinely used to detect HIV suspected AIDS patient, HIV suspected AIDS patient, HIV HIV in suspected AIDS patient. PCR is now routinely used to detect HIV in suspected AIDS patient. HIV in suspected AIDS patient. It is being used to detect suspected mutations in gene in suspected cancer patient. To detect mutation in gene in suspected cancer patient. It is powerful technique to identify many other genetic disorder. It is powerful DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. PCR. Polymerase chain reaction. Detect. Diagnose. By HIV in suspected AIDS patient detects HIV suspected AIDS patient. You can say HIV detectors detect HIV in suspected AIDS patient. Detects mutations in suspected cancer patient. Okay. It is a technique to identify many genetic disorders. It is powerful technique to identify many genetic disorders. It is powerful technique to identify many genetic disorders. It is powerful technique to It is a powerful technique to identify many genetic disorders. It is powerful technique to It is powerful technique. It is powerful technique to identify many other genetic disorders. It is powerful technique to identify many other genetic disorders. It is powerful technique to identify many other genetic disorders. It is powerful technique. It is powerful technique to It is powerful technique to detect. It is powerful technique to identify or detect. Identify many Powerful technique to identify many genetic disorder. Powerful technique to detect, identify. Okay, ELISA is based on principle of okay. ये तो है ना यहाँ देखिए. It is powerful technique to identify the genetic disorder. Identify many other genetic disorder. Genetic disorder PCR. Genetic disorder PCR. Genetic disorder PCR. Okay. A single-stranded DNA or RNA tagged with a radioactive molecule radioactive molecule. A single-stranded DNA or RNA tagged with a radioactive probe. Okay, this is probe. This is DNA or RNA. A single-stranded DNA and or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA. Okay, hybridize होगा. Dies to its complementary DNA. Hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. A single-stranded DNA or RNA tagged to the radioactive molecule probe is allowed to hybridize to its complementary DNA. Its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film. because the probe will not have complementarity with the mutated gene. Okay. A single-stranded DNA or RNA tagged with a single-stranded DNA or RNA tagged with a radioactive probe. Radioactive. Probe. Okay, this is probe. Radioactive molecule probe. DNA. Hybridized to its complementary DNA in a clone of cell followed by detection using autoradiography. Is allowed to hybridize to its complementary DNA in a clone of cells followed by detection using autoradiography. Single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA is allowed to hybridize with its complementary DNA in a clone of cell using by detection using autoradiography. A molecular probe might be used to find the nucleotide sequence. Okay, DNA in a clone cell followed by detection using autoradiography. Autoradiography, photographic film. Followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. The clone having the mutated gene will hence not appear. Clone. Gene. Mutated gene. Clone with mutated gene. Clone with mutated gene. Tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cell, followed by detection using autoradiography. A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene. Clone with mutated gene do not appear on photographic film. Because the probe will not appear because not appear on photographic film because probe will not have complementarity with the mutated gene. This is because probe will not have this is because probe will not have complementary with the mutated gene, not appear on photographic film. This is because not have complementary with mutated gene. ELISA is based on the principle of antigen-antibody interaction. ELISA, enzyme-linked immunosorbent assay, is based on principle of antigen and antibody reaction. Based on the principle of antigen-antibody reaction interaction. Antigen-antibody interaction. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens. वाईफाई चली गई, लाइट चली गई ना. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins. Antigens are proteins or glycoproteins, etc. Or by detecting the antibody synthesized against the pathogen. Antibody synthesized against the pathogen. Identify specific protein. ELISA. Excuse me. Enzyme-linked immunosorbent assay. ELISA is based on the principle of antigen antibody interaction. Based on the principle of antigen antibody interaction. Identify some protein. Based on the principle of antigen antibody interaction. Did identify some protein. Glycoprotein. Infected cell secretes antibody. These are based on principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigen. Protein or glycoprotein. By detecting the antibody synthesized against the pathogen, or by detecting the antibody, infected cell secretes antibody. Infection can be detected. Infection by pathogen can be detected. Infection by pathogen can be detected by 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 the presence of antigen. Infection by pathogen can be detected by the presence of antigens. Infection by pathogen can be detected by the presence of antigens. Protein or glycoprotein. Protein or glycoprotein. ELISA is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of Antigens. What's the Kerala? They must have laughed at me. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigen, protein and glycoprotein, etc., or by detecting the antibody synthesized against the pathogen, or by detecting the antibodies synthesized against pathogen. You ELISA को principle antigen-antibody, okay, interaction, okay. Recombinant DNA को cancer. RNA na bakili number sa uta DNA single stranded, okay, single stranded. It's not probe log also. This is probe. Probe is radioactive molecule. Generate complementary hybrid DNA. Do not express a clone with mutated gene. Do not express gene. Do not express in photographic film. Autoradiography. A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA. In a clone of cell, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementary. Do not appear in photographic film because probe will not have complementary. Probe will not have complementary with mutated gene because probe will not have complementary with mutated gene. Good luck, Alex. Thanks, Pawan Joshi. Hi, Mukesh. मैं चैट्स ऑफ कर दूं? एलेक्स, आजकल तुम ना मुझे क्लोवर लीफ नहीं देते हो, सिर्फ गुड लक लिखते हो. प्रोब विल नॉट, मैंने मेंशन भी किया था. हाँ, बस 2 ही दिया क्योंकि मुझे पता है तुम्हें भी एग्जाम में चाहिए ना. मैं चैट्स ऑफ कर दूं? प्रोब विल नॉट हैव कॉम्प्लीमेंट्री विद म्यूटेटेड. Probe will not have complementary with mutated gene. Probe will not have complementary with mutated gene. Transgenic animals, animals that have had their DNA manipulation to possess and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced. Animals that have had their DNA manipulated to possess and express. Transgenic animals, animals that have had their DNA manipulated to possess and express an extra foreign. Animals that have had their DNA manipulated to possess and express an extra foreign genes are known as transgenic animals. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced. Although over 95% of all existing transgenic animals are mice, why are these animals being produced? How can man benefit from such modification? Let's try and explore some of the common reasons. Transgenic animals, animals that have had their DNA manipulated to possess and express an extra foreign gene. DNA manipulated and express an extra foreign gene. DNA manipulation. Animals that have had DNA manipulation to possess, express an extra foreign gene are known as transgenic animals. Animals that have had their DNA manipulated to possess and express an extra foreign gene. Animals animals that have their animals that have had their gene DNA animals that have had their DNA manipulated animals that have had their DNA manipulated animals that have had their DNA manipulated to possess and express an extra foreign gene to possess and express an extra foreign gene to possess and express and extra. Okay, animals that have had their DNA manipulated to possess and express an extra foreign genes are known as transgenic animals. Transgenic animals. Foreign gene. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Okay, 95% transgenic animals are mice. 95% transgenic animals are mice. For existing transgenic animals are mice. Okay, transgenic rats, rabbits, pigs, sheep, cow, and fish. Pigs. Normal physiology and development, study of disease. Normal physiology and development, study of disease. Biological products, vaccine safety, chemical safety testing, vaccine safety, chemical safety testing, vaccine safety, biological products, study of disease. Study of disease, vaccine safety, chemical testing safety, chemical safety testing, vaccine safety, biological products, study of disease, and normal physiological and development, normal physiology and development. Chemical safety testing. Okay, chemical safety testing. That is chemical safety testing. Along with vaccine safety. Study disease. To study disease. Normal physiological PHY. Physiology, normal physiology and development. Vaccine testing, safety testing, chemical safety testing, study of disease. Study of disease. Study of disease. हाँ तुम कर सकते हो, लेकिन तुम करना मत, ओके? घर पढ़ के दिया। हाँ कर ली भाई, देख रहे हो विनोद। That was embarrassing though. कनिका सेफ्टी, मैं ऑकवर्ड फील करूंगी, ऐसा मत करो ना तुम लोग। कनिका सेफ्टी टेस्टिंग, वैक्सीन सेफ्टी, स्टडी ऑफ डिजीज, नॉर्मल फिजियोलॉजी एंड डेवलपमेंट, बायोलॉजिकल प्रोडक्ट्स। Biological products, chemical safety testing, vaccine safety, study of disease, normal physiology and development, biological products, vaccine safety, biological product, vaccine safety, chemical safety testing, chemical safety testing, chemical safety testing, vaccine safety, chemical safety testing, biological products, normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example, study of complex factors involved in growth such as insulin, like growth factor. Study of complex factor involved in growth such as insulin like growth factor by introducing genes from other species that alter the formation of this factor and studying the biological effects that result by introducing genes from other that result. Information is obtained about the biological role of the factor in the body. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example, the study of complex factor involved in growth such as insulin like growth factor by introducing genes from other species that alter the formation of this factor and studying the biological effects that result. Information is obtained about the biological role of the factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as model for human disease, so that investigation of new treatment for disease is made possible. Today, transgenic model exists in many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Cystic fibrosis, Alzheimer, and rheumatoid arthritis. Cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as model for human disease, so that investigations of new treatment for disease is made possible. Today, Transgenic models exist in many Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Model. Transgenic model. Model human. Transgenic model for many human disease. Transgenic model for many human disease exist. What is it? Transgenic model for many human disease exist. EXIST exist. Alzheimer, rheumatoid arthritis, Alzheimer, rheumatoid arthritis. Alzheimer, rheumatoid arthritis, and मैं भूल गई. Cystic fibrosis and cancer. Cystic fibrosis and cancer. Cystic fibrosis, cancer, rheumatoid arthritis, Alzheimer, ALZH, Alzheimer. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contributed to the development of disease. These are specially made to serve as models for human disease so that investigation of new treatment for disease is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, Alzheimer, and rheumatoid arthritis. Normal. This is a study of disease, okay? First one with you, normal physiology and development. Normal physiology and development, study of disease. Transgenic animals का role. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example, study of complex factor involves in involved in growth, such as insulin-like growth factor, by introducing genes from other species that alter the formation of that alters the formation of this factor and studying the biological by introducing this gene from other species that alter the formation of this factor and studying the biological effect that result. Information is obtained about the biological role of the factor in the body, normal physiology and development. Transgenic animals role, normal physiology and development, study of disease. Normal physiology and development, study of disease. Biological products, medicines required to treat certain human disease can contain biological products. Medicine required to treat certain human disease can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA or genes which codes for the particular product, such as human protein alpha 1 antitrypsin, human protein alpha 1 antitrypsin, alpha 1 antitrypsin used to treat emphysema. Treatment alpha 1 antitrypsin, human protein alpha 1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of PKU, phenylketonuria, and cystic fibrosis. In 1997, the first transgenic cow, Rose, produced human protein enriched milk. 2.4 gram per liter. Human protein enriched milk, 2.4 gram per liter. The milk contains the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Alpha 1 antitrypsin, human protein alpha 1 antitrypsin, treat emphysema. Human protein alpha 1 antitrypsin, 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 alpha 1 antitrypsin, alpha 1 antitrypsin, human protein alpha 1 antitrypsin used to treat emphysema. PKU, phenylketonuria, PKU, and cystic fibrosis. The first transgenic cow, Rose, produced human protein enriched milk. 2.4 grams per liter. 2.4 grams per liter. The milk contained the human alpha lactalbumin, human alpha lactalbumin, and was nutritionally a more balanced product for human babies than natural cow milk. This is biological products. Biological products. Human alpha 1 trypsin. Human alpha 1 trypsin biological products. Human alpha 1 trypsin treat emphysema. Emphysema. Human alpha 1 trypsin treat emphysema. Biological product. Human alpha 1 trypsin treat emphysema. Human alpha 1 trypsin treat emphysema. Biological product. The other one is emphysema treat phenylketonuria, PKU. Phenylketonuria, and nuria, PKU. Phenylketonuria, PKU. Okay. And cystic fibrosis. Then number cystic fibrosis. CYS, cystic fibrosis. FIBR versus cystic fibrosis, phenylketonuria, human alpha-1 trypsin, treat emphysema, human alpha-1 antitrypsin, human alpha-1 antitrypsin used to treat emphysema, human protein, okay, human protein, alpha-1 trypsin, human protein, alpha-1 trypsin, treat emphysema. Similarly, attempts are being made for treatment of phenylketonuria, PKU, and cystic fibrosis. 1997, transgenic cow. In 1997, transgenic, TRANS, transgenic cow. Name Rose, 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 2.4 gram per liter. 4 gram per liter है ना. Human protein, milk protein, alpha-lactalbumin, transgenic cow, Rose, 2.4 gram per liter, human milk protein, alpha-lactalbumin. The milk contained the human alpha-lactalbumin. Okay. The first transgenic cow, Rose, produced human protein enriched milk, 2.4 gram per liter. Attempts have been made for treatment of phenylketonuria, PKU, and cystic fibrosis. In 1997, the first transgenic cow, Rose, produced human protein enriched milk, 2.4 grams per liter. The first transgenic cow, Rose, produced human protein enriched milk, 2.4 grams per liter. The milk contained the human alpha-lactalbumin. Human protein enriched milk. Emphysema triggers the human protein alpha-1 trypsin. Alpha-1 trypsin treat emphysema. Alpha-1 trypsin, okay. Phenylketonuria र cystic fibrosis को लागि पनि सेम कुरा गर्नु परेको छ. Phenylketonuria, cystic fibrosis को लागि, okay. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccine. Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Okay. Mice are being used to test the safety of polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Chemical safety testing. This is known as toxicity safety testing. Okay, chemical safety testing, toxicity safety testing. Chemical safety testing like toxicity safety testing. The procedure is same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. Okay, transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain result in less time. Toxicity testing in such animals will allow us to obtain result in less time. Okay. Normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example, study of complex factor involved in growth, such as insulin-like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects that result. Information is obtained about the biological role of the factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as models for human disease, so that investigations of new treatments for disease is made possible. Today, transgenic model exists for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Today, transgenic model exists for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Okay, first one is another natural physiology and development. Natural physiology and development and study of disease, biological production is the biological products and the key vaccine safety, chemical safety testing, Alzheimer, rheumatoid arthritis, cystic fibrosis, and cancer. Biological products ma emphysema treat garcha human protein alpha 1 trypsin, okay. त्यहाँदेखि सेम स्टेप्स दिएको छ पिकेयु, फिनाइल किटोनुरिया र पिकेयु र के को लागि लिएको छ आई थिंक क्यान्सर ना सिस्टिक फाइब्रोसिस, फिनाइल किटोनुरिया एन्ड सिस्टिक फाइब्रोसिस, ओके. ट्रान्सजेनिक काउ 1997 हालैको कुरा जस्तो 97 मा ट्रान्सजेनिक काउ रोजे यसले के गर्यो ह्युमन प्रोटिन इनरिच मिल्क प्रड्युस गर्यो 2.4 ग्राम पर लिटर थियो. ह्युमन अल्फा लैक्ट एल्ब्युमिन जो कि ह्युमनको बेबीजको लागि मोर सुइटेबल थियो एनिमल्सको भन्दाखेरि. भ्याक्सिन सेफ्टी माइसमा गर्छ. पोलियो भ्याक्सिनको सेफ्टी पनि गरेको थियो. फर्स्ट माइसमा गर्छ देन मङ्कीमा गर्छ कि. टेस्टिङ टक्सिसिटी सेफ्टी टेस्टिङ, केमिकल सेफ्टी टेस्टिङलाई के भन्छ टक्सिसिटी सेफ्टी टेस्टिङ पनि भन्छ. Biological products. Medicines required to treat certain human disease can contain biological products, but such products are often expensive to biological products are often expensive to make. Transgenic animals that produce useful biological products can be created. Transgenic mice that produce useful biological products can be created by the interaction of the portion of DNA or genes which codes for a particular product, such as human protein alpha 1 trypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria, PKU, and cystic fibrosis. In 1997, the first transgenic cow, Rose, produced human protein-enriched milk, 2.4 grams per liter. The milk contained the human alpha-lacto albumin and was nutritionally a more balanced product for human babies than natural cow milk. Vaccine safety. Transgenic mice are being developed for the use of testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Chemical safety testing. This is known as toxicity safety testing. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to toxic substances. And the effects studied. The toxicity testing in such animals will allow us to obtain result in less time. ethical issue. The manipulation of living organism by the human race cannot go on any further. The manipulation of living organism by the human race cannot go on any further without regulation. Some ethical standards are required to evaluate the morality of all humans. Okay, some ethical standards are required to evaluate the morality of all human activities that might help or harm living organism. Ethical standards are required to evaluate the morality of all human activities that might help or harm living organism. Morality of all human activities that might help or harm living organisms. Going beyond the morality of such issues, the biological classification of such things is also important. Going beyond the morality of such issues, the biological significance of such thing is also important. Going beyond the morality of such issues, the biological classification of such thing is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Genetic modification of organisms can have unpredictable results. Therefore, the Indian government has set up organizations such as GEAC, Genetic Engineering Approval Committee, GEAC, 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 Genetically Engineered Approval Committee, Genetic Engineering 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 Approval Committee in India. Okay, the Indian government has set up organizations such as the GEAC, Genetic Engineering Approval Committee, Genetic Engineering Approval Committee, Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and the safety of introducing GM organisms for public service. Okay, which will make decisions regarding the validity of GM research, and the safety of introducing GM organisms. Okay, make decisions regarding the validity of GM research and the safety of introducing GM organisms or public service. Which will make decisions regarding the validity of GM research and the safety of introducing genetically modified organisms for public service, which will make decisions regarding the validity of GM, genetically modified research, introducing GM organisms for public service. Okay. The modification uses of living organisms for public service as food and medicine source, for example, has also created problems with patent granted for the same. The modification uses of living organism for public service. The modification uses of living organism for public service. कितना अंधेरा दिख रहा है ना, लेकिन लाइट भी तो क्या ही कर सकते, वो आ ही नहीं आ रहा है लाइट. Therefore, Indian government has set up organisms such as Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and the safety of introducing GM organisms for public service. The modification uses of living organisms for public service as food and medicine. The modification uses of living organisms for public service as food and medicine resources, for example, has also created problems with patent. The modification uses of living organisms for public service as food and medicine sources. For example, has also created problems with patent granted for the same. There is growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic materials. Plants and other biological resources that have long been identified, developed, and used by farmers and indigenous people of a specific region, country. मैं तो दिख भी नहीं रही हूँ ठीक से, वाह, लेकिन कोई बात नहीं, 2 मिनट ही और पढ़ना है. Rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. Rice is an important food grain, the presence of which goes back Rice is an important food grain, the presence of which goes back thousands of years in. Okay, rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. Nice. Rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. There are an estimated 1 tens, 100,000, 10,000, lakh, 2 lakh varieties of rice in India. 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and flavor. And 27 documented varieties of basmati are grown in India. 2 lakh varieties of rice in India, richest. Diversity of rice in India, richest in the world. Slot 1 completed. Okay, basmati rice is distinct for its unique aroma and flavor. 27 documented varieties of basmati are grown in India. There is reference to basmati in ancient text, folklore, and poetry, as it has been grown for centuries. In 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. The new variety of basmati had actually been derived from Indian farmers' variety. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent uses, products, and process based on Indian traditional herbal medicine. Example, turmeric, neem. If we are not vigilant and we do not immediately counter these patent applications, other countries, individuals may encash on our rich legacy and we may not be able to do anything about it. Okay. 10 मिनट का ये लिए लाइट. Yep. आई ये लो 10 मिनट का ब्रेक. अभी तो मैं नहीं टाइप कर सकती ना यहाँ से, तुम लोग ही टाइप करो. हाँ, मैं पिन कर दूंगी. कंटिन्यू करते हैं विदाउट ब्रेक. क्यों? बोलो भाई साहब, क्यों ऐसा क्यों? फ्लो रहेगा, ओके। फिर कंटिन्यू करते हैं। स्लॉट 2 लिख दो, स्लॉट 2 60 मिनट लिख दो तुम, अल्पिन। आई थिंक आज लाइट नहीं आएगा, ये इतना ज़्यादा बारिश होती है देखना। सेकंड स्लॉट स्टार्ट कर रहे हैं, ओके। लेट मी स्ट्रेच फर्स्ट। मुझे कुछ भी दिखाई नहीं दे रहा है तो ठीक से, लेकिन गुड लक एवरीवन। आई थिंक ये स्लॉट मैं कंप्लीट नहीं कर पाऊंगी, ओके, क्योंकि ना मुझे नहीं दिखाई देगा, ये ख़त्म होगा इसका चार्ज जल्दी ही। Biopiracy is a term used to refer the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Biopiracy is the term used to refer to the use of bioresources by multinational companies, MNCs. Okay. Biopiracy. Use of bioresources by MNC and other organizations. Use of bioresources by MNC and other organization. Use of bioresources by MNC and other organization without proper authorization from the countries and the people concerned without compensatory payment. Okay, biopiracy. Use of bioresources by MNC and other organization. Use of bioresources by MNC and other organizations without proper authorization from the countries and the people associated with without proper authorization from the countries and the people concerned without compensatory payment. Biopiracy. Okay, Okay, 27 documented varieties of basmati. 27 documented varieties of basmati. 1997 मा अमेरिकन कंपनीले के गर्यो पेटेन्ट राइट्स पायो बासमती राइसमा थ्रु यूएस पेटेन्ट एन्ड ट्रेडमार्क अफिसबाट. ओके, crossed with semi-dwarf varieties. Indian basmati was crossed with semi-dwarf variety. Okay, 2 lakh variety of rice का पाउँछ इन्डियामा पाउँछ. The diversity of rice in India. Herbal medicine को सब पनि सेम गरेको छ. Most of the industrialized nations are rich financially but poor in biodiversity. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to the bioresources can be exploited to traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, efforts and expenditure during their commercialization. Good luck, Nishant. Most of the industrialized nations are rich financially but poor in the biodiversity and traditional knowledge. In contrast, the developing and underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure during their commercialization. Okay. There has been growing realization of the injustice, inadequate compensation, and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patents Bill. That takes such issues into consideration, including patent terms, emergency provision, and research. Patent terms, emergency provision, and research, and development initiative, research, emergency provision. Patent terms, emergency provision, research, and development initiative. लगभग हो ही गया है ये चैप्टर. Okay, Agrobacterium Agrobacterium vector थियो. Vector जस्तै युज गरे Agrobacterium. Okay, कहाँनिर लेखेको छ भने यहाँदेखि agrochemical based agriculture मा युज गर्छ. Okay, यहाँदेखि पढौँ. 3 options that can be thought for increasing food production. Agrochemical based agriculture, organic agriculture, and the genetically engineered crop based agriculture. Okay, तीनटा छ. Increase production of food increase गर्नको लागि agrochemical based agriculture, त्यहाँदेखि organic agriculture, and genetically engineered crop based agriculture. Green revolution. टिस्यू कल्चरमा के हुन्छ, एउटा टेक्नोलोजी हो या फिर प्रोसेस हो जहाँनिर एक्सप्लान्ट गरेर होल नयाँ जुनै पनि होल प्लान्ट रिजेनरेट गर्न सक्छ. 1950 मा फर्स्ट टाइम गरेको थियो साइन्टिस्टहरूले के ड्युरिङ 1950s. Whole plant could be regenerated from explants. Okay, a totipotency के हो, capacity to generate whole new plant from explant. Totipotency. Tissue culture, totipotency. Then micropropagation, method of producing thousands of plants through tissue culture is called micropropagation. Okay, genetically identical हुन्छ original plants. Okay, like even soma clones, they are genetically identical soma clones. It is important to stress here that nutrient medium must provide a carbon source such as sucrose and also inorganic salt, vitamins, amino acid and growth regulator like auxin and cytokinin. Nutrient medium मा के चाहिन्छ carbon source जस्तै sucrose, vitamins, amino acids and cytokinin र auxin, plant growth regulator. Inorganic salts पनि चाहिन्छ. Okay, carbon source, inorganic salts, vitamins, amino acid and growth regulator. Okay, पाँचवटा चीज चाहिन्छ. Method of producing thousands of plant micropropagation, genetically identical to original plant हुन्छ. Soma clones भन्छ त्यसै कारण. Soma clones. Okay, majority of food plants like tomato, banana and apple. Tomato, banana and apple etc. produce गरेको छ. Okay, Soma clones. Okay. Another important recovery of healthy plants from diseased plant, meristem, apical meristem and axillary meristem of infected plant बाट recover गर्न सक्छ. Okay, in vitro हुन्छ virus free plant बनिन्छ. Meristems of banana, sugarcane, potato and potato तीनटा कुराको कल्चर गरेको तीनटा कुरालाई कल्चर गरेको छ. ओके, इन्फेक्टेड प्लान्टबाट डिजिज फ्री प्लान्ट बनाउनु पर्यो. Potato, sugarcane and banana. Isolated single cell from plants and after digesting the cell wall have been able to isolate naked protoplast surrounded by plasma membrane. Isolated protoplast from two different varieties of plant, each having a desirable character, can be fused to get hybrid protoplast. Hybrid protoplast लाई भन्छ somatic protoplast. Somatic. Yes, somatic protoplast भन्छ somatic hybrid भन्छ, which can be further grown to form a new plant. These hybrids are called somatic hybrid. यो process लाई के भन्छ? Somatic hybridization. Somatic hybridization भन्छ process लाई. All the best for your exam, brother. If not, we'll get the result. Thanks, Vaibhav. Get hybrid protoplast, which can be further grown to form a new plant. These hybrids are called somatic hybrid, while the process is called somatic hybridization. Imagine a situation when a protoplast of tomato is fused with that of potato. Protoplast of tomato is fused with that of potato. They are grown to form new hybrid plant combining tomato and potato characteristics. Well, this has been achieved resulting in the formation of potato, potato and tomato. Process Q, somatic hybridization. Somatic hybrid. Tomato Q, somatic hybrid. Process like somatic hybridization. Unfortunately, this plant did not have all the desired combination of characteristics for its commercial utilization. Use of genetically modified crop is a possible solution. Use of genetically modified crop is a possible solution. Plant, bacteria, fungi and animals whose genes have been altered by manipulations are called genetically modified organisms, GM. Genetically modified organisms, GM. Genetically modified plants have been used in many ways. Genetic modification has made crop more tolerant to abiotic stress, cold, drought, salt, heat. Tolerant to abiotic stress. Made crop more tolerant to abiotic high comfort zone. Let's study, okay? Plant bacteria. मुझे ये ऑफ कर देना चाहिए ना, मेरे सामने ही चार्ट्स खुला है तो मैं पढ़ पा रही हूँ अच्छे से. Now I can't read the charts, okay? मैं हाइड कर रही हूँ खुद के लिए. Okay, कहाँ थी मैं? Reduce reliance on chemical pesticides. Pest-resistant crop. Made crop more tolerant to abiotic stress, cold, drought, salt, heat. reduced reliance on chemical pesticides. Pest-resistant crops help to reduce post-harvest help to reduce post-harvest losses, increased efficiency of mineral uses by plant. This prevent early exhaustion of fertility of soil. Increase efficiency of mineral uses by plants. This prevent early exhaustion of fertility of soil. Okay, pest resistant crop. Okay, help to reduce post harvest losses, increased efficiency of mineral uses by plant. This prevent early exhaustion of fertility of soil. Increased efficiency of mineral uses by plants. This prevent early exhaustion of fertility of soil. Enhance nutritional value of food. Example: golden rice, that is vitamin A enriched rice. Enhance nutritional value of food. Golden rice, vitamin E. Example: golden rice, that is vitamin A enriched rice. In addition to this uses, GM has been used to create tailor-made plant to supply alternative resources to industries in the form of starches, fuels, and pharmaceuticals. Some of the application of biotechnology in agriculture Some of the application of biotechnology in agriculture that you will study in detail are the products of pest-resistant plant which could decrease the amount of pesticide used. Bt toxin is produced by a bacterium called Bacillus thuringiensis, Bt for short. The production of pest-resistant plant which could decrease the amount of pesticides used. Bt toxin is produced by bacterium called Bacillus thuringiensis. Bt toxin genes has been cloned from the bacteria and been expressed in plants to provide resistance to insect without the need for insecticides. In effect, created the biopesticides. Bt cotton, corn, potato and tomato, along with rice and soya bean. Bt cotton, Bt corn, rice, tomato, potato and soya bean. Bt corn, rice, tomato, potato and soya bean. Rice, soya bean, tomato, potato. Bt corn. Bacillus thuringiensis. Okay, Bt cotton. Some strains of Bacillus thuringiensis produce proteins that kill certain insects such as Lepidopterans. Armyworm and cotton. Tobacco budworm and armyworm. Coleopterans, beetles, and dipterans, flies, mosquitoes. Lepidopterans, tobacco bodyworm, dipterans, and coleopterans. Bacillus thuringiensis form protein crystal during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Why does this toxin not kill the bacillus? Actually, the Bt toxin protein exists in inactive protoxins. But once an insect ingests the inactive toxin, it is converted into active form of toxin due to the alkaline pH of the gut, which solubilize the crystal. The activated toxin binds to the surface of the midgut epithelial cell and create a pore that cause cell swelling and lysis and eventually cause the death of the insect. Okay. Cell swelling and lysis. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. The choice of genes depends upon the crop and the targeted pest. Specific Bt cotton genes were isolated from Bacillus thuringiensis and incorporated into several croplands such as cotton. No, I can't do that. Specific Bt cotton. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several croplands such as cotton. The choice of genes depends upon the crop and the target pest, as most Bt toxins are insect group specific. Bt toxins insect group specific. The toxin is coded by a gene Cry1AB. Cry1AB, Cry, okay. Cry1AB and Cry2AC. Cry2AB yenira. Cry1AC unse yenira. Cry1AC. Cry1AC and cotton bollworm. CO51. Cotton bollworm. Bollworm. Cry1AC ra Cry1AB. Cry1AB corn borer. C-O-R-N corn borer. Corn borer. Cry1AB controls corn borer. Cry1AC and Cry2AB. Cry1AC and Cry2AB control the cotton bollworms. That of Cry1AB controls corn borer. There are a number of them. For example, the protein encoded by the genes Cry1AC and Cry2AB control the cotton bollworm. That of Cry1AB controls corn borer. Cry1AC nahi na? Yeah, Cry1AC Cry. Named Cry. There are a number of them. The example protein encoded by the gene Cry1AC and Cry2AB. Cry1AC and Cry2AB cotton bollworm control the cotton bollworm. That of Cry1AB controls corn borer. Pest-resistant plant. Okay, pest-resistant plant. Several nematode parasite, a wide variety of plant and animal, including human being. A nematode, Melodygyne incognitia, infect the root of tobacco plant and cause. Okay, Melodygyne incognitia nematode. Okay, infect root of tobacco plant. Root of tobacco plant and causes a great reduction in yield. A novel strategy was adopted to prevent this infestations, which was based on the process of RNA interference. RNA interference. RNAi. It's like RNA silencing takes place in all eukaryotic organisms as method of cellular defense. This method involves silencing of a specific mRNA due to complementary double-stranded RNA. This method involves silencing of a specific mRNA due to complementary double-stranded RNA molecule that binds to and prevent translation of mRNA. Silencing. The source of this complementary RNA could be from an infection by virus having RNA genome or mobile genetic element. Transposomes. Mobile genetic element. Transposomes that replicate via an RNA intermediate. Using agrobacterium vector, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA. Agrobacterium vector, nematode-specific. Agrobacterium vector. Nematode-specific genes were introduced into the host plant. मैंने बोला था पीसी डेड होगा. मैं चलो दुकान जाती हूँ, कि दुकान से ना लेके आती हूँ. मैं पॉज कर लेती हूँ तब तक टाइमर. कैंडल के नाम पे एक तो सही बचा है, टुकड़ा, एक टुकड़ा बचा है। थोड़ा थोड़ा तो दिख रहा है ना। ये स्लॉट ना ऐसे ही कंप्लीट कर लेते हैं, ओके। उसके बाद मैं दुकान जाऊंगी और बड़ा बड़ा कैंडल लेके आऊंगी, फिर एक बार में 3 कैंडल जलाएंगे। Agrobacterium vector. Nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in host cells. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in host cells. I think मेरा वन आई तो पेन करने लगा, तो क्या हो गया? इतना भी हाई स्ट्रेंथ नहीं दिया मैंने। आई ड्रॉप भी अंदर नहीं बाहर निकल के आया। Using agrobacterium vector, nematode-specific genes were introduced into the host plant. Agrobacterium vector. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. The two RNA being complementary to each other form a double-stranded RNA that initiate RNA interference and thus silence the specific mRNA of the nematode. The consequences was that the parasite could not survive in a transgenic host expressing a specific interfering RNA. The transgenic plant therefore got itself protected from parasite. Molecular diagnosis स्टार्ट किया ना, ये दो करना फिर। Recombinant DNA technology made immense impact area of healthcare. Safe and more effective therapeutic drugs. Unwanted immunological response as is common in case of similar products isolated from non-human resources. अरे भाई साहब ये बाल को ना। At present, about 30 recombinant therapeutics have been approved for human use in India. 12 in India, 12 of this presently being marketed. 12, okay, 30 total, 30 in India, 12 total, 30 India में 12 मत है. Genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at regular time interval. Okay. Insulin used for diabetes was earlier extracted from the pancreas of slaughtered pigs and cattle. Slaughtered cattle and pigs extracted from pancreas of, okay. Allergy and other reaction, type of reaction. Two short polypeptide chain, chain A and chain B, that are linked together by disulfide bridge. In mammals, including human, insulin is synthesized as prohormone, Like proenzyme, the pro hormone also needs to be processed before it becomes a fully mature and functional hormone. Okay. Extra stretch C peptide, not present in mature insulin, removed during maturation C peptide. Okay. The main challenge for production of insulin using rDNA, recombinant DNA technology, was getting insulin assembled into mature DNA. Ali Lilly, American company, 1983. The American company prepared two DNA sequence corresponding to A and B. Two DNA sequence corresponding to A and B chains of human insulin and introduced them in plasmid. Ali Lilly, American company. DNA sequence corresponds to A and B chain of human insulin and introduced them in plasmid of E. coli to produce insulin chain. Chain A and B separately extracted and combined, creating disulfide bonds to form human insulin. Gene therapy, hereditary disease, can be corrected. Corrector therapy. Gene therapy is an attempt to correct hereditary disease. Gene therapy, collection of method, allows correction of gene defect diagnosed in child embryo. Genes inserted in person's cells or tissue to treat disease. Delivery of normal gene into individual or embryo to take over the function of and compensate for the non-functional gene. First clinical gene therapy. First clinical gene therapy was given in 1990. 4-year-old girl, 1994-year-old girl, ADA deficiency, adenosine deaminase deficiency, adenosine deaminase deficiency. Enzyme replacement therapy, bone marrow transplantation. Enzyme replacement therapy, bone marrow transplantation. Deletion of gene for adenosine deaminase. 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 Okay. A deletion of gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. Bone marrow transplantation. Bone marrow transplantation by enzyme replacement therapy. In some children, ADA deficiency can be cured by bone marrow transplantation, enzyme replacement therapy. Okay, enzyme replacement therapy. Okay, in which functional ADA is given to the patient by injection. But the problem with both of these approaches is that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patients are grown in a culture outside the body of functional ADA cDNA using a retroviral vector cDNA, a functional ADA cDNA, okay, cDNA, circular DNA, using a retroviral vector is then introduced into the lymphocyte, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the genes isolated from the marrow cells producing ADA is introduced into cells. Shut up. Gene isolated from marrow cells producing ADA is introduced into cells at embryo cells. Okay, introduced into cell. Genes isolate from marrow cell producing ADA is introduced into cell at early embryonic stages. It could be permanent cure. हो गया ये वाला तो बायोटेक्नोलॉजी वाला करना पड़ेगा ना. सबसे पहले ये कर लेते हैं ना. Where is it? बायोटिक का 2 चैप्टर में 1 चैप्टर ही क्या. Can do it again if needed or required. Microbes and human welfare, okay. Beside microscopic plants and animals, microbes are the major component of biological system on this earth. You have studied the diversity of living organisms in class 11. Do you remember what kingdom among the living organisms contain microorganisms, Monera and fungi, I guess? Fungi is microorganism? No, just Monera and Protist, I guess. Which are, yeah, Monera and Protist. Which are the one that are only microscopic. Microbes are present everywhere in soil, water, air, inside our bodies, and that of other animals and plants. They are present even at sites where no other life form could possibly exist. Sites such as the inside the geyser, thermal vents, where the temperature may be as high as 100 degrees Celsius deep in the soil. 100 degrees snow. Several meter thick, and in highly acidic environment. Microbes are diverse protozoa, bacteria, fungi. Oh my, fungi भी तो है microbes देखो. Protozoa, bacteria, fungi, and microscopic animals and plant viruses. Microscopic animals and plant viruses. नींद आ रही है, नहीं आ रही है नींद? पढ़ो. And also prions that are proteinaceous infectious agent. Okay. Microbes are protozoans, bacteria, fungi, microscopic animals and plant viruses. Protozoa, bacteria, fungi, microscopic animal, plant viruses, viroids, and also prions that are proteinaceous infectious agent. Prions are proteinaceous infectious agent. Proteinaceous infectious agent. Proteinaceous infectious agent. Proteinaceous infectious agent. 4 बार तो कर ही सकते हैं, मिनिमम 5 बार तो कर ही सकते हैं ना. एक का ही 5 बार किया है. प्लांट किंगडम. Microscopic animals and plant viruses, viroids, and also prions that are proteinaceous infectious agents. Some of the microbes are shown in figure 8.1 and 8.2. Microbes are like bacteria, and many fungi can be grown on nutritive. Okay, microbes like bacteria, and many fungi can be grown on nutritive media to form colonies that can be seen with the naked eyes. With the naked eyes. Microbes like bacteria and मैंने कल टेस्ट नहीं दिया, बुद्धू लड़की को देना था, तो मैं भूल गई ना. It's okay. Microbes like bacteria and many fungi can be grown on nutritive media to form colonies that can be seen with the naked eyes. Such cultures are useful in studies on microbes. Bacteria, rod-shaped bacteria magnified 1500 times. Okay, rod-shaped bacteria rod-shaped magnified 1500 times. Spherical shaped magnified 1500 times. A rod-shaped bacterium showing flagella magnified 50,000 times. Okay, 1500 to 50,000. Rod-shaped bacterium flagella. A rod-shaped bacterium showing flagella. Okay. Head, collar, tail, plate, pins and prongs. Pins, plate, tail, collar and head. Head, collar, tail, plate, pins and prongs. This is This is bacteriophage and this is tobacco mosaic virus. First one is bacteriophage, second one is tobacco mosaic virus, TMV. TMV ये नीचे वाला है भाई साहब, मैं हमेशा क्यों गलती करती हूँ. TMV, compact rod shaped virus. Okay, bacteriophage and adenovirus which cause a respiratory infection. Okay, this is adenovirus which cause respiratory infection. 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 Adenovirus cause respiratory infection. Adenovirus cause respiratory infection. एक तो बैक्टीरियोफाज हो गया और ये वाला TMV, टोबैको मोजाइक वायरस. Rod shaped TMV, tobacco mosaic virus, magnified about 1 lakh to 1,50,000 times. 1 lakh to 1,50,000 times. Okay. Compact rod shaped virus, TMV. Okay. This is adenovirus, respiratory infection. Adenovirus. Respiratory infection. Bacteriophage. Rod-shaped tobacco mosaic virus. Okay. This is bacterial colony. This is fungal colony. Bacterial colony. Okay. Colonies of bacteria growing in petri dish. Fungal colony growing in petri dish. Colonies of bacteria growing in petri dish. Colonies of fungi growing in petri dish. Adenovirus causes respiratory infection. Adenovirus causes respiratory infection. Adenovirus causes respiratory infection. पहले बायो तो कंप्लीट कर लेना इसका, बाकी का देख लेंगे फिर. Compact rod shaped virus. Okay. अभी भी बहुत कुछ करना है. ये एक बार तो कर लिया है ना, एक बार से ज़्यादा क्या है इस बार. Proteinaceous infection. Prions are proteinaceous infectious agent. Okay. Diverse protozoans, bacteria, fungi, microscopic animals, and plant viruses and viroids. Okay. Major component of biological ecosystem on Earth. Microscopic plant and animals. Okay, ये वाला adenovirus, respiratory infection, TMV. Basilak है. You have read the microbes. Fermented beverages, antibiotics, chemical enzyme and other bioactive molecules. यहाँ से वो आएगा ना, सीवेज ट्रीटमेंट यहाँ से था. In chapter 7, you have read the microbes cause a large number of diseases in human beings. They also cause disease in animals and plants. In chapter 7, you have read that growth and development. Okay, एक और बात करना पड़ेगा फिर. Read that microbes cause a large number of diseases in human beings. In chapter 7, you have read that microbes cause a large number of diseases in human beings. In chapter 7, you have read that microbes cause a large number of diseases in human beings. They also cause disease in animals and plants. But this should not make you think that all microbes are harmful. Several microbes are useful to man in diverse ways. In chapter 7, you have read that microbes cause a large number of diseases in human beings. You'll be fine, Nina. You'll be fine. You'll be fine. They also cause disease in animals, plants, but this should not make you think that all microbes are harmful. Several microbes are useful to man in diverse ways. Some of the most important contributions of microbes to human welfare are discussed here in this chapter. Microbes and household products. You would be surprised to know that we use microbes or products derived from them every day. You would be surprised to know that we use microbes or products derived from them every day. A common example is the production of curd from milk. Microorganisms such as lactobacillus and other commonly known as lactobacillus, lab, something acidy, I forgot. Lactic acid bacteria. Lactic acid bacteria. Yeah. Lactobacillus and other commonly known as lactobacillus. Not lactobacillus, it's lactic acid bacillus. Lactic acid bacillus. Lactic acid bacteria, not bacillus. Ah, God. Lactobacillus. Lactic acid bacteria. Lactic acid bacteria. Lab. Grows in milk and convert it to curd. Lactic acid bacteria. Lactic acid bacteria. Lactobacillus, lactic acid bacteria. Okay. Convert it to curd. During growth, the lab produce acid that coagulate and partially digest the milk protein. A small amount of curd added to the fresh milk as inoculum or starter contain millions of lab, which at suitable temperature multiply, thus converting milk to curd, which also improves the nutritional quality of okay, which also improves its nutritional quality by increasing vitamin B12. A small amount of curd added to the fresh milk as inoculum or starter contain millions of lab, which at suitable temperature, which at suitable which at suitable temperature multiply, thus converting milk to curd, which also improves the nutritional quality of increasing which also improves the nutritional quality by increasing vitamin B12. Lactic acid bacteria lab and the lactobacillus. That's it. यति मात्रै याद गर्नु छ यो प्याराग्राफबाट अगाडिको बकवास दिएको छ. In our stomach too, the lab plays very beneficial role in checking disease causing microbes. Okay. In stomach, lab plays a beneficial role checking disease causing microbes. Checking disease causing microbes. मुझे नहीं लिखना चाहिए, मेरा बुक गंदा हो जाएगा. Lab plays a very beneficial role in checking disease causing bacteria. हाम्रो भुँडीमा के गर्छ, ल्याबले disease causing microorganisms लाई के गर्छ, पिक गर्छ या फिर डिटेक्ट गर्छ. Stomach, lab plays very beneficial role in checking disease causing microbes. In checking disease, okay, check गर्दो रहेछ, मार्दिन्छ होला सायद, लिमिटमा राख्नुको लागि. Checking disease causing microbes. Vitamin B12. कर्डमा के बढ्छ, vitamin B12. दुईटा bacteria छ, lactobacillus and lactic acid bacteria. Vitamin B12 enrich हुन्छ in our stomach, के गर्छ, disease causing microbes लाई check गर्छ. The dough which is used for making foods such as dosa and idli is also fermented by bacteria. The puffed up appearance of dough is due to the production of CO2 gas. Can you tell which metabolic pathway is taking place, resulting in the formation of CO2? Where do you think the bacteria for this fermentation come from? Similarly, the dough which is used for making bread is fermented using baker's yeast. Dosa and idli is also fermented by bacteria. Am I visible? Anyone? Am I visible? Am I visible? अगर मैं visible हूँ तो please लिख दो. Thank you, Baba. लेट्स स्टार्ट. ओके, अभी भी 13 मिनट रिमेनिंग है ये स्लॉट के लिए. मैंने टाइमर बस पॉज किया. Aditya, I'm kind of embarrassed, okay, but thank you so much. You're the one who paid, right? I don't know why you paid, but still okay. It's embarrassing. शर्म भी आ रही है. Alex, I got money. चलो पढ़ते हैं. Embarrassing लग रहा है, but it's okay. Thank you, Aditya. फर्स्ट टाइम है ना, इसलिए मुझे थोड़ा शर्म लग रहा है, वरना नहीं लगता. प्लीज ऐसा मत बोलो ना, मुझे एम्बैरेसिंग लग रहा है. मैं फेस ही नहीं कर पाऊंगी ना कैमरा को. पता नहीं कौन थे, लेकिन किसी ने भेजा है. ओके, बट स्टिल आई एम काइंड ऑफ एम्बैरेस्ड, यू नो. है ना एलेक्स? मुझे इतना एम्बैरेसिंग क्यों लग रहा है? पहले तो मुझे नहीं लगा था कभी भी इतना एम्बैरेसिंग. इट्स ओके माँ, इट्स ओके, इट्स ओके माँ, इट्स ओके. ऑक्सीजन लो. हाँ लूंगी लूंगी ऑब्वियसली. चलो पढ़ते हो. ओके, मैं टाइमर स्टार्ट करूँ? मैं भी खुश हूँ, लेकिन मुझे एम्बैरेसिंग लग रहा है, पता नहीं क्यों. बोला तो दिस इज फर्स्ट टाइम ना, इसलिए इसलिए शायद एम्बैरेसिंग लगा होगा. ओके, गुड लक. स्टार्ट करूँ? टाइमर 13 मिनट बचा है सेकंड स्लॉट का. तो तुम्हें भेज दूँ? वो तो अभी मैं भी नहीं निकाल पाती, नहीं निकाल सकती. इसका भी कितना सारा रूल्स होता है, मुझे नहीं पता था. मुझे भी थोड़ा थोड़ा ही पता चल रहा है. ओके, गुड लक एवरीवन. 13 मिनट ही. ओके. The puffed up appearance of dough is due to the production of CO2 gas. Can you tell which metabolic pathway is taking place resulting in the formation of CO2? Where do you think the bacteria for this fermentation come from? The dough which is used for making food such as the puffed up appearance of dough is due to the production of CO2 gas. Can you tell me which metabolic pathway is taking place resulting in the formation of CO2? Where do you think the bacteria of this fermentation come from? Similarly, the dough, which is used for making bread, is fermented using baker's yeast, Saccharomyces cerevisiae. A number of traditional drinks and foods are also made by fermentation by the microbes. A number of traditional drinks and foods are also made by fermentation by the microbes. Toddy, a traditional drink of some part of southern India, is made by fermenting sap from palm. Toddy, a traditional drink of some part of southern India, is made by fermenting sap from palms. Microbes are also used to ferment fish, soya bean, bamboo shoot, and make food. Okay. Is made by fermenting sap from palms. is made by fermenting sap from palms. Is made by fermenting sap from palms. Toddy, a traditional drink of some part of southern India, is made by fermenting sap. Made by fermenting sap from palms. Sap from palms. Toddy, a traditional drink of some part of southern India, is made by fermenting sap. Fermenting sap from palm. Toddy, a traditional drink of some part of southern India, is made by fermenting sap from palms. Fermenting sap. palm को सापलाई के गर्नुपर्छ, ferment गर्नुपर्छ, तब के बनिन्छ, टोडी बनिन्छ. Microbes are also used to ferment fish, soya bean and bamboo shoot to make foods. Cheese is one of the oldest food items in which microbes were used. Cheese is one of the oldest food. Different varieties of cheese are known by their characteristic texture, flavor and taste. Their specificity coming from the microbes used. For example, the large holes in Swiss cheese are due to production of large amount of CO2 by a bacterium named Propionibacterium shermani. For example, the large hole in Swiss cheese are due to production of a large amount of CO2 by a bacterium named Propionibacterium shermani. Swiss cheese, okay. Large amount of CO2 production, large holes. Swiss cheese, large amount of CO2. Propionibacterium shermani. Propionibacterium shermani. Propionibacterium shermani. Swiss cheese, उसले बनाउँछ bacterium हुन्छ, एउटा bacterium छ, Propionibacterium shermani. The Roquefort cheese are ripened by growing a specific fungi on them. Fungi, okay. Roquefort cheese, fungi. FR. For real. Which gives them a particular flavor. Which gives them a particular flavor. For real. FR. Fungi, Roquefort, okay. Bacterium. Penicillium Roquefort for Roquefort cheese. बफर को फिक्स ही करना पड़ता है. अगर फिक्स नहीं हुआ या फिर ना लैग जब कर रहा है, फिक्स नहीं किया तो खुद ही एंड हो जाता है लाइव सेशन. आई थिंक अब मैं विजिबल हूँ शायद. अभी भी विजिबल नहीं हूँ. अब फिर से जाएगा, ये स्टेबल तो नहीं रहेगा, इतना तो पता है.